All right, so here we're going to talk about an introduction to medical genetics. So we're going to give some background and some history, talk a little bit about some diseases and how genetics has evolved over time. We'll talk about the different types of genetic diseases, such as chromosome, single gene disorders, etc. And then we'll also discuss what is a true genetic disease versus what is a multifactorial disease that has also environmental influences. Okay, so we already have talked about DNA, RNA, so we should be very clear on what the difference between an intron versus an exon is. Um, and then we're also going to talk more about phenotype and genotype. We have dominant versus recessive transmission patterns, a homozygote which carries two copies, identical copies of the gene, and a heterozygote who does not. And a diploid gene is normal gene cells in our body that have two copies of each chromosome versus a haploid, which are the sex chromosomes, sperm and eggs, that have only one chromosome each. We'll also go into eye color and describe different genes that control eye color, so it's not as simple as a Mendelian inheritance pattern. And then talk about different disorders and how they are inherited. So first off, why is this important? It's important because we are learning more and more about how genes influence phenotypes. And some of this are things that we can see, such as eye color, hair color, but others are things that have to do with how our body functions. And so if we're able to detect a deleterious genotype before an infant is born, this can help to prepare the parents. Alternatively, we can also do genetic testing now to see if there are certain genes that run in families that can be potentially passed on to the next generation, which can be informative to parents as they are going to start a family. So in the 1900s, we rediscovered Mendelian laws, and we also have evolved from there to do screening for certain genetic disorders, such as Tay-Sachs disease. Screening started in the 1970s. We also started amniocentesis for chromosome disorders in the 1970s. We have some tests that can be for certain diseases, such as Huntington disease that runs in families, so we can do a predictive genetic testing for this disease. And we also, of course, in 2001, the Genome Project, we have our full human genome that has been sequenced. And now we're moving towards things like gene editing with CRISPR technology. Huntington's disease may be a target for CRISPR. And of course, there always is the idea of designer babies. You want your hair color, eye color chosen. And hopefully the CRISPR technology will be more for correcting disorders. So if you think about the history of genetics, in the first edition of the Mendelian Inheritance for Man, which was published in 1368, listed 1368 autosomal disorders, and about 119 X-linked disorders. And today we have 21,000 autosomal disorders and over 1,000 X-linked. There are also some Y-linked disorders, though a bit more rare, and mitochondria disorders, which have maternal inheritance patterns, and those are also very rare. So there has been some knowledge of genetics playing a role in phenotypes, especially with respect to flies, fruit flies, as well as plants and farming. The Human Genome Project was initiated in the 90s. It completed in the 2000s, and we now have a complete sequence, of course, of the human DNA. And so there is a goal to understand the relationship of genes, their structure, and to the phenotype, of course, and how variation contributes to phenotype and diseases. Now, what is a gene? A gene is essentially a sequence in your DNA that is required to give a code for making an amino acid sequence of a protein. So the DNA sequences are going to be composed of introns, exons, and also different control elements such as promoter regions and enhancer regions, etc. Now the exons are the protein coding regions. And so if we look at our gene here, we can see we have exon 1 and exon 2 and exon 3. And in between the exons, we have introns. So these introns are intervening sequences, and these are not part of the protein that will eventually be expressed. So genes are going to be variable in sizes. The average is about 3,000 base pairs. The largest is 2.4 million base pairs. The sequence are going to be separated by intervening sequences, and we're going to have on our DNA a template strand, which is going to be the fidelity of the DNA and carries the biological information of making the proteins necessary for our life. So basically, a gene is a unit of inheritance. You want to make sure that your genes are not mutated when you pass them on to the next generation. Johannesson is given credit for coining the word gene in 1909. Okay, so let's break this down a little bit. So here we have alleles. Alleles are going to be genes, and they are going to be different versions of the same genes. So here you see we have an allele that has a big B, and in the same location on the second chromosome, we have an allele of the same gene, but it has a little b. So this is for eye color. So the big B stands for brown and the little b stands for blue. All right, so how you can view this is we have this cassette tape. Many of you may not remember cassette tapes, but cassette tapes were used for songs. But the tape is a recorded information. So recorded information is similar to our DNA. We record this, we want to make sure it stays intact, it doesn't get altered. Now the cassette is a large bundle of these recordings, so you can think of this as a chromosome. And so on every single chromosome, there is a lot of recorded gene information. One song or one track on this tape is similar to a gene. It's part of that chromosome. And different versions of the song sung by different people are like alleles. And so we have our one chromosome, which are here, and we have our DNA, and we have our different alleles, which are our big B, and we have our little b. Now, phenotype is what we actually see, or is the function of whatever that protein is. So in the case of visual, we can see eyes and hair color very easily. We can see colors of flowers very easily. So those are the physical appearances of the genes presenting in this phenotype. A genotype is a combination of alleles for a particular gene. So here we have a genotype of big B, big B. Here we have a genotype of big B, little b. And here we have a genotype of little b, little b. And so here we have a homozygote, has the same allele, big B, big B. Here we have a heterozygote, big B, little b, and here is another homozygote. Now, with respect to heterozygotes, there are dominance and recessive. So a dominant allele is going to be always expressed when it's present, meaning that you cannot mask it by another allele. And so in this case of brown eyes, brown is a dominant allele over blue. So even if a blue allele is present, the individual will present with a brown-eyed phenotype. Recessive alleles is an allele which can be masked by another allele, and therefore it's only going to be expressed when two copies are present. So in this case, you have a homozygote blue eye, so it gets presented as blue eyes because brown is not present to dominate over the blue. Now, if an individual has two copies of the same allele, this is a homozygote. So we have a BB or a BB, and both are homozygote. And a heterozygote is at the same gene is different on the two chromosomes, so this is going to be a heterozygote. Hetero means other, homo means same. Now, a cell which contains two copies of each chromosome is called a diploid, and all the cells in our body are going to be diploid, except for our germline cells, sperm and eggs. So they're going to contain two chromosomes, one maternal and one paternal. A cell which contains one copy is haploid, and these are sex cells. These are all going to be haploid. Sperms, eggs, pollen, plant egg cells, etc. Those are all going to be haploid. our gametes are going to be produced by meiosis, and this is where you have your diploid parent cell shown here, and then the chromosomes get copied shown here. So now you have 2 copies of your brown. So here's your, let's see, B, and 2 of your blue, little b, little b. So this is a heterozygote. And then each of these 4 chromosomes are going to be put into an individual sperm in this case. So this one will have a little blue, a little b, and these will both be large b. But each of the sperm only carry 1 copy of the chromosome. Eggs are also produced the same way. Again, we have a heterozygote, B, B. They get duplicated, 2 browns, 2 blues, and then each of the eggs now have 1 copy of each eye color. And this is the process of meiosis. Now in fertilization, you're going to get 2 of the gametes, a sperm and an egg, that come together to form a diploid zygote. And so again, we have our large B here and our little b here. And so this zygote is going to be a heterozygous for a brown and a blue eye. And the process is random. So the zygote could have gotten a blue eye and a blue eye, or a brown and a brown. So inheritance of eye color is going to be blue versus brown. So in this case, egg with a little b fused with a sperm with a little b, and so we're going to have a phenotype of BB, which is going to be blue. And in the opposite case, this is large B, so this is going to be brown. Very straightforward. And remember that brown is always going to be dominant, and so if you have a heterozygote, you're going to get brown. Okay, so if it's so straightforward, why do we have so many shades of color? So why do we have green eyes? Why do we have light blue? Why do we have hazel eyes? Well, there's more to it than just this brown and blue color. Even though brown is still going to be dominant, there are other genes that are influenced. So we have some genes that have been identified since the Genome Project. On chromosome 15, there's a gene called OCA2. Now there are 2 forms. One is brown, which is dominant over blue. So in a person who has brown eyes, the OCA2 gene is produced, has high melanin. And in a person who has blue eyes, the OCA2 gene that is not produced, and this leads to low melanin. So melanin is adding color to the eyes to generate brown, and this is driven by OCA2 gene. Now there is a second gene. This gene is called GAY, or G-E-Y, and this is on chromosome 19. Now there are 2 forms of this gene, green and blue, and green is going to be dominant over blue. So in the case of a green GAY gene, you have some melanin produced, and a blue GAY gene, you have no melanin produced. And so the influence of both the GAY gene as well as the OCT2 gene is going to influence the different variation in colors of eyes that people have. All right, so now let's talk briefly about, just in general, the different chromosome or the different types of genetic diseases. So we're going to go through each of these individually. We're going to go through chromosome disorders in general, single gene disorders, multifactorial, and mitochondria disorders. And so let's start first with our chromosome disorders. So for our chromosome disorders, we're going to have the chromosome be altered. And you can have this alteration as a missing chromosome, so you can have completely a lost chromosome, or you can have some part duplicated, deleted. Chromosome deletions is going to be part of the chromosome is missing or deleted, and this is thought to be caused by errors during crossover, during meiosis. You can also have duplication, too much information. You have duplication of an entire chromosome, or you have duplications of sections of chromosomes. And so with duplication, you get a different copy number. So with duplication, you get a different copy number of genes within that area of a chromosome. So here we have single chromosome mutations. The first is a deletion. And so you can see that this section is deleted, so this chromosome is truncated, shorter. And then for duplication, which is number 2, this chromosome is elongated and it has additional information. And then for inversion, you basically are flipping. So you have A, B, C, and this becomes C, B, A. So the inversion will flip a certain area. Now when 2 chromosomes are involved, so this is just a single chromosome over here. When you have 2 chromosomes involved, you can have an insertion where you have chromosome 1, and it adds genes to chromosome 2. So chromosome 1 is now truncated, and chromosome number 2 is elongated. Or you can have a translocation. And a translocation is going to be where you take a piece of each chromosome and they swap. And so these chromosomes keep their size. However, a gene or sections of the genes have moved from one chromosome to another, so they flipped or swapped information. The next step are single gene disorders. Okay, so in our single gene disorders, this is going to be caused by variations or mutations in a single gene, sickle cell anemia, for example. Okay, and so the DNA changes are going to affect the product of the genes, so protein affected. And it really depends on what the protein is as to the phenotype. So the feature of each disorder is related to the specific gene that is affected and the function of that protein in the actual body. Some of these genetic disorders are very serious, and children who have them are extremely sick and often cannot survive after birth. Others are relatively easy to manage, sickle cell anemia, for example, with proper care, and most people nowadays have full lives with these types of disorders. And it's well known that the chances of a good outcome increase if the condition is identified before or soon after birth. Now, they are caused by a single gene variations, and so some of these are going to run in families. When one or both of the parents are carriers of a genetic disorder, they have a chance of passing it on to their children, even if they themselves are healthy. So up here is a pedigree generation map, and you can see that we have the different generations, generation 1 through generation 4. And they're coded, so our dark codes are for the affected males, and our dots are for our carrier females. So we can see that in generation 1, we have a carrier female that passed the carrier trait to 2 of its daughters, and one daughter did not get the carrier trait. So in this side, we have no affected males. Now, of the ones who had a carrier trait, we can see that we have one affected male, another carrier, and another affected male. And here we have a carrier and another affected male. And in the primary, we also have an affected male. And so this is going to be a X-linked recessive pattern of inheritance. And it's X-linked because only the mothers are transmitting it. And none of the daughters are getting it. So that means that in order for a daughter to get it, they have to have 2 copies of the gene. The males only need 1 copy because they only get 1 X chromosome. So the chance of a person passing on this genetic disorder to their children depends on the disease as well as the inheritance pattern. So again, this one is an X-linked recessive. So the chance of them passing it on to their male offspring is 50%. And the chance of a female being a carrier is also 50%. So if someone knows that they have a single gene disorder in their family, they can have genetic testing to see if they have those causing variations. So over here in this green side, where the mother was, or the daughter was not a carrier, this line is unaffected. The remainder are affected. So if a male in this or a female wanted to know if they're a carrier, they can just go and get genetic testing. Now, some cases, single gene disorders are going to be caused by a nouveau mutation. De novo mutation, which is new. And this means that there was no family history of having this disorder, but it arose during the formation of the eggs and sperm at some point during the chromosomes being replicated. And so this new mutation is now introduced into the family and can be passed on to subsequent generations. But all previous family members would not have a history of this particular gene. Okay, so next we're going to talk about multifactorial disorders. Multifactorial disorders are when there is one factor causing a trait or health problem, such as a birth defect or chronic illness. And the main factor are genes, but the cause includes other factors that aren't genes. And these factors could be things like nutrition, environment, cigarette smoke, etc. Some of the more common multifactorial disorders are schizophrenia, diabetes, asthma, depression. High blood pressures, Alzheimer's, obesity, heart diseases, etc. So there are many of them. And so in general, you're going to have some sort of combination of gene input and environment input involved. So within this little diagram here, we have some diseases that are influenced only by genes. Down syndrome, for example. Down syndrome is a duplication of a chromosome, and so you have too much information. And some are going to be strictly environment. We have our infections, our microbes. And then others are going to have both gene and environment influences, such as this bubble over here. And finally, we have mitochondria disorders. So our mitochondria disorders are a group of conditions that affect the mitochondria. And people who present with these conditions can present at any age with almost any affected body system. However, most of the affected systems will include the brain, muscles, heart, liver, nerves, eyes, ears, and kidneys. Symptoms of severity are going to vary widely. Mitochondria disorders can be caused by changes or mutations in the mitochondria DNA or nuclear DNA. Those caused by mutations in mitochondrial DNA are always transmitted by maternal inheritance. So for those who are nuclear DNA, can either come from the mother or the father. All mitochondria come from the mother. The sperm does not give any mitochondria for the growing fetus. Now, treatment is going to vary on the type of condition and the signs and symptoms of each person. So in this case here, we're showing maternal inheritance of our mitochondria. So we have an affected mother here, and the affected mother is going to give the mitochondria to all their offspring. And so all their offspring will be affected. And then individuals also the offspring of the affected will also be affected. You can show here. All the females that continue to have children will keep infecting or affecting their offspring. So this one has all of them. Now here we have a male that's affected, but the male does not pass on any mitochondria, so none of the females down here are affected. So in essence, the mitochondria inheritance disorder has been eliminated from that branch of the family tree, as well as this branch of the family tree as well. Okay, so we do have different patterns of inheritance. We have Mendelian pattern of inheritance, and this is going to follow what we are usually think about with genetics. We have the autosomal dominant, autosomal recessive, X-linked recessive, and X-linked dominant. Dominant always means if you have a copy of the gene that's affected, you're going to present with the phenotype. Autosomal means that they're non-sex chromosomes, and X-linked is obviously going to be the X chromosome. And so for an autosomal dominant, only one copy of the mutated gene is required to present with the phenotype. Autosomal recessive, you're going to need both copies mutated. So autosomal recessive tends to run in families with consanguinity. Consanguinity is a term used for cousins or very close relatives having children together. X-linked recessive, you're going to need 2 copies required for the female, 1 in the male. And so you're going to see mostly a mother-to-son inheritance pattern with the daughters being carriers. X-linked dominant is you're going to have 1 copy mutated is required for both male and female. And then Y-linked, you're always going to see a father-to-son inheritance pattern. Now the non-Mendelian are other things that can influence phenotypes that don't follow these patterns. We have something called imprinting, in which the imprinted gene is turned off. So that means that in every individual that has a normal phenotype, they only have 1 copy that's turned on, and it has to be non-mutated. If you have this copy that's turned on and does get mutated, then typically you're going to have some sort of disease because the other copy on the other chromosome is shut off by imprinting. We also have just mitochondrial inheritance pattern, which we just spoke about, and that's very rare. And so this is because only the mother can give mitochondria from the egg to the growing fetus. Multifactorial are going to be both environment and genetic influence. We have sporadic or de novo, so that's not inherited but it arises. And we have continuous gene syndromes, and this is where you have a chromosome abnormality in which you have several genes that are in close proximity or either duplicated or deleted. And this can cause multifactorial disease presentations because in this region of the genome, for example, you might have an effect on metabolism, and you also might have an effect on bone growth, for example. 2 different groups of genes, but since they're in the same area that was affected, they're going to have the presentation of the phenotype will affect both. All right, so again, we have our environmental caused diseases, our infectious diseases, influenza. We have our true genetic diseases, cystic fibrosis, hemophilia, and then we have our multifactorial diseases that have influences both from the genetic and from the environment.